Okay, so DNA and protein synthesis. Question number 1 says, what is the primary function of DNA? Now, the primary function of DNA is to store those hereditary instructions for making proteins. Store the hereditary or genetic, that works, so genetic information for making proteins. Primary function of DNA is to store the genetic or the hereditary information for making proteins. Okay, number 2 says one strand of the DNA has the following bases. What would the complementary strand be? So we're doing complementary DNA this time. This process is called replication. We're doing DNA replication. We're going DNA to DNA. G would go with C. T would go with A. This is DNA, so A will go with T. C with G. A with T. C with G. T with A. C, G, T, A, T, A, C, G, T, A, C, G, T, A. Yeah, we're good. Okay, number 3 says GAT is on a strand of DNA. What would be the mRNA strand? So we're going from DNA template to the formation of the mRNA strand. So G, we go with C. A would go with U this time because it's RNA, and RNA has uracil instead of thymine. So A goes with U, and T goes with A. Number 4 says list 3 ways in which DNA differs from RNA, or RNA differs from DNA. So careful, this question will most likely be on your test. Pay close attention to it. It says, how do they differ? It may give you 3 facts about it, but if one of them's not different, then that's not the correct answer. So this is how RNA differs from DNA. Well, first we just talked about it. We know that RNA contains uracil instead of thymine. RNA contains uracil instead of thymine. You put U's in RNA instead of T's. And another way is RNA is single-stranded, while DNA is double-stranded. DNA looks like a twisted ladder. RNA looks like a ladder who's been cut right down the middle of the rungs. You should see like Tom and Jerry. He'd go boom, boom, boom down through the ladder, split it in half. Well, that's what RNA looks like. Half of that ladder. Okay, the third way that RNA is different from DNA is RNA can leave the nucleus. DNA cannot. That's why we have to have that mRNA. RNA can leave the nucleus. DNA cannot. Number 5 says a process in which a molecule of DNA is copied into a complementary strand of mRNA is called, we're going DNA to mRNA, that is called transcription. It's that first step in protein synthesis. They happen in alphabetical order. Transcription happens first. It's transcription. Now I'm going to put a little note here beside transcription just to help us remember. Put it in red so we notice it. This is just to summarize it that we have DNA to mRNA. That equals transcription. And since the DNA cannot leave the nucleus, this is happening in the nucleus, which is also the answer to this blank. This occurs in the nucleus. Okay. So if we're making mRNA, it's transcription and it happens in the nucleus. Then the mRNA carries the coded DNA message from the nucleus out to the, well, it's carrying it to where the protein is going to be made, and proteins are made by the ribosomes, so it's carrying it out to the ribosomes. Okay, pay close attention to number 7. I'm pretty sure you will see this one on the test as well. It might even be almost word for word. 7 says the process of transcription can be summarized as, well, a key word in this question is transcription. What's happening in transcription? We just said transcription is going from the DNA to the mRNA. Keyword transcription. So we are making mRNA during transcription. Okay. These all start with the formation of. Well, if we're forming something, that means we're making it. So this is like saying we're making what? Transcription, we're making mRNA. So eliminate some answers here. A says the formation of DNA. Well, that's not correct, so we can eliminate A. B says the formation of a protein using a DNA template. We're not making a protein in transcription, so A and B have been eliminated. We're making mRNA. Transcription's making RNA, so it's the formation of RNA. So these two both start out correct. The formation of RNA. On transcription, we're making that mRNA from our DNA template. So the formation of RNA, an RNA molecule using a DNA template would be our correct answer. This one said using a protein template. No, we're using the RNA to make the protein, not the other way around. So C would be our correct answer. Transcription is the formation of a DNA, of an RNA molecule, using a DNA template. Formation of an RNA molecule using a DNA template. Okay, this funny looking little thing down here is kind of like little notes I put up here. This is called the Central Dogma. I'm not sure why it's called the Central Dogma. That's just what the scientists called it. So we'll put that there. You might possibly see a question that says, this represents the what? This represents the Central Dogma. Dogma is just a way of thinking about something. So we use DNA to make our DNA template to make our mRNA. From our mRNA, we are going to make, our ultimate goal is to make a protein. So that X right there is representing the protein. I'm going to put it right there because that's what it should look like. It should say DNA to RNA to protein. That's the short version of protein synthesis, DNA to RNA to protein. So what best replaces that X? The X is best replaced by protein. Number 9 says a codon is a sequence of 3 nucleotides that codes for an amino acid. If the DNA sequence shown below goes through transcription into an mRNA and translation into a protein, how many amino acids will result from this sequence? And well, we said it takes 3 bases to make a codon for 1 amino acid. So to do this, we can go, put those in. We can count 1, 2, 3, draw a line. And we can go 1, 2, 3, draw a line. 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 So we're going to group them into groups of 3 because it takes groups of 3 to be able to code for an amino acid. So we have 1, 2, 3, 4, 5, 6 groups of 3. So this would code for 6 amino acids. And sometimes they might ask you the other way. If there are 6 amino acids in a polypeptide, how many bases were in the DNA template? Well, then you know there were 3 bases for each of those 6, so you would just say 6 times 3 equals 18 bases. So it goes either way. If you're given a big number of how many amino acids, you're going to divide by 3. If you're given the amino acids and it wants to know how many bases or nucleotides, then you're going to say amino acids times 3. Number 10, ribosomes attach to the mRNA. Ribosomes decode the message and carry out the formation of protein. This process is called, well, this is the final step of protein synthesis. The mRNA code is translated into a protein. So this is called translation. Transcription and translation. 2 steps. Transcription comes first in ABC order. It happens first. C comes before L. The structure of DNA, that double helix, that twisted ladder structure, was discovered in 1953 by 2 gentlemen by the name of Mr. Watson and Mr. Crick. So Watson and Crick discovered that structure. Number 12 says proteins are macromolecules that are built from various combinations of, well, the building blocks of proteins are amino acids. So it's those combinations of amino acids that make the proteins. Various combinations of amino acids. Number 13 says, which bases are classified as pyrimidines? This is where we said Y's go together. There's a Y in the word pyrimidine. So we're going to pick our bases that also have a Y. And that would be thymine and cytosine. So our pyrimidines are thymine and cytosine. Thymine and cytosine. Y's go together. There's a Y in pyrimidine. There's a Y right there in thymine. And there's a Y right there in cytosine. The other 2 would be purines. Adenine and guanine are purines. And then thymine and cytosine are pyrimidines. Number 14 says, which part of the DNA molecule provides the code for the proteins? Our molecules are nucleotides, have sugars, phosphates, and bases. That's the 3 main parts of the nucleotide. 3 main parts of the DNA molecule. It's the bases that provide the code for making those proteins, the bases. Adenine, guanine, thymine, and cytosine. Those are what are going to code for the proteins. Number 15 says, which codon serves as the starter for protein synthesis? We talked about that a few minutes ago. School starts in August. So AUG is our start codon. It codes for the amino acid methionine. So that would be stored in that protein. Number 16 says, list the 3 types of RNA. The first one we talked about was mRNA. And that is messenger RNA. And its function is it carries the DNA message or DNA instructions for protein synthesis from the nucleus to the ribosome. mRNA, messenger. I'll do this again. Okay, then we had our tRNA. That's the transfer RNA, or I like to call it the tow truck RNA. And it picks up the amino acids